Well, hi everyone. It's my great pleasure to provide a bit of introduction for Dr. Francis Collins, our next keynote speaker. And you know, as I was thinking about this morning, I thought, well, I can go through all the sort of perfunctory things like he has a PhD from Yale in physical chemistry and an MD from the University of North Carolina and so on and so forth. And I thought, well, that's got to be really boring. Maybe I should just sort of give a quick tour through a few aspects of Francis Collins that you may or may not know about. So I'll be brief, but I thought this might be of interest in terms of an introduction. So who is Francis Collins? Francis Collins, you can meet the man here who led the team that mapped the human genome and sequenced it too, by the way. He and I started actually on this project at the same time in 1993, so I feel like we have a shared history, but now I'll go into detail about ways that Francis and I differ, just in case you're interested. So I'm not the director of the NIH, but Francis is. You can see here the offer being made to him by Barack Obama as he's working in the laboratory, obviously setting up a new microscope, I believe, was the topic of the conversation, and obviously he accepted the job. Francis, unlike me, will appear in pictures with children and Muppets, and also with dogs. He is a rock star of science, and you'll see evidence of that, I think, at the end of his talk, and then later on this evening after we've had a bit of drink and lobster. And I'm also, unfortunately, was not able to get Joe Perry to accept my invitation to co-appear with Francis, so you'll have to just have Francis. He's a biker. So many people don't know this. I'm close to being a biker, having paid for a Harley Davidson that I occasionally get to ride on the back of, but that's about as close as I come in that comparison. And then last but not least, he taught Stephen Colbert about the Brain Initiative just recently, where Stephen, intrigued as always by props, asked this important question. However, I don't think that Francis should be held responsible for Colbert's interpretation of the Brain Initiative, shown here in comparison to the gun registry. And then I will just finish by saying that, in my opinion, Francis is a visionary leader. He's shown here with Barack Obama again, having just announced the Precision Medicine Initiative and co-published with Harold Varmus in the New England Journal of Medicine about the importance of this initiative. And I'll finish there by saying that I think he'll tell us a lot more about that in his remarks now. So please welcome Francis. That was okay.
Well, thanks, Elaine. That was unexpected and generally quite kind. So I'm sure you had a lot of material you decided not to use, and thank you for that, too. It's a great privilege to be here at Cold Spring Harbor and to follow such an elegant presentation by George Davey Smith. And I want to tell you a little bit about precision medicine, but first I can't help but reflect, at least briefly, on the things that have happened over the course of the last many years, where many people have gathered in this room and other rooms like this on this property to see what could be done on this grand adventure of understanding the human genome, because it has been quite a ride. And I've been fortunate to be here for many of these gatherings. If you go and look on my shelf in my office there in the director's office, you'll notice there's something up there on the top shelf. I just decided to blow that up so you can see, well, yeah, there was a bunch of times that I was here with those abstract books. I'm sure you're saving yours too. But you'll notice it kind of went to 2011 and then stopped. And so this is the first time I've been able to come back to this meeting now for the last 4 years. And it's just joyful to be back here again, seeing some of the science and seeing some of my old friends and seeing how this field is so vigorously alive. But the Genome Project had much of its history conducted at Cold Spring Harbor in the meetings like this one, where people got together to try to figure out how to make something really challenging happen. And there were moments there that are unforgettable. Jim Watson is sitting in the front row, so I'm reminded of 1998 when there was a bit of a challenge to the public project by a private entrepreneur. And there were great anxieties about whether this was going to lead to a bad outcome. And Jim, at breakfast one morning in Blackford, as this was all playing out, challenged me whether I intended to play Winston Churchill or Neville Chamberlain. you can imagine what that felt like. Yeah, that was a time. It all turned out extremely well. And may I say it turned out well because of our international partners, because of the willingness of the 2,400 scientists who worked on the Genome Project to give all the data away every 24 hours initially. And for that ethic to have now infused itself into this field of genomics is an enormous contribution to the way in which this science has gone forward and has become really the norm for many other fields as well, who have learned from the field of genomics just how empowering it is to be someone who favors public access in every possible way. And I've had the chance in this very property, that's a bit of a blurry picture, but that was a picture taken at Cold Spring Harbor near the end of our wild ride, trying to finish the sequence of the human genome. And you will recognize probably quite a few of the people in that picture who were the leaders of this international effort that ultimately in 2003 announced for the first time that we had finished the human genome. And we've had several more announcements of finishing since then, and we're not finished with finishing, so be prepared for more of that. But one thing I will say is that with all of this excitement, there are a few people who, looking at that kind of finishing announcement, would say, okay, well, we did the genome. We are now in the post-genome era. No, we are not in the post-genome era. We spent all of human history in the pre-genome era, doggone it, and now we finally have the genome. We're in the genome era, and let's not rush through it. We might get there someday, but we're certainly not there now. Otherwise, why are we here? If we're in the post-genome era, we should go to some other meeting. So what has happened since, of course, the genome sequence itself, which was challenging, but in many ways was less complicated than what we're trying to do now, which is to understand function, is a whole lot of other projects have come along to try to help these poor healthcare providers here who are having trouble understanding what to do with G's, T's, A's, and C's. And all of us working together over what has been a pretty interesting decade or more of adding information to the genome have done so with projects like HapMap, 1,000 Genomes, ENCODE, goodness, a whole lot of different kinds of organized efforts, some of which are not all that obviously directly related to genomics, but everything is connected. There's even something here called BARD. That's the Bioassay Research Database, which gives me a chance to quote from Hamlet, there are more things in heaven and earth, Horatio, than are dreamt of in your philosophy. I think there are more genome research projects than are dreamt of in our philosophy 12 years ago. And isn't that a very good thing? So I just want to give you a quick snapshot of a few things that from now my perspective as NIH director give me great sense of excitement and enthusiasm about the way in which this field has blossomed in many directions that I don't think most of us necessarily would have anticipated 12 years ago. The ability to use genomics to really take apart the problem of cancer and to look at the molecular level in thousands, tens of thousands of tumors to really begin to build the catalog of how good cells go bad. Most recently in the squamous cell carcinoma publication, the Cancer Genome Atlas and their international partners doing a lot of this work. There's a paper soon to come out in Cell on melanoma with similar kinds of exciting findings. When you have lots of tumors, you learn lots of information. Over and over again, we emphasize that power is all about numbers. The microbiome project, which I think was considered to be kind of an interesting investigation when we started it a few years ago, knowing that we could use DNA sequencing to identify microbes that we couldn't culture in the laboratory. The way in which this has found its way into subjects such as obesity and diabetes and maybe even autism is really a remarkable and unexpected development and has empowered a whole field that I think is really taking off. The epigenome, the ability across the genome to be able to assess what's happening in terms of chromatin being open or closed and how that relates to gene expression is very heavily represented at this meeting, and well, it should be because of the way in which these tools and technologies built upon DNA sequencing in many instances have made it possible for us to really take apart at a pretty impressive level the program of how cells differ from each other and how differentiation occurs. My own laboratory in the intramural program at NIH has primarily now focused on epigenomics. And we've had a great good time, in fact, studying this in the case of diabetes, both in terms of human pancreatic islets and human muscle. That's a photograph of folks from my lab who are here at the meeting and who were presenting the 4 posters that you see here. I hope some of you had a chance to browse and look at those. I should say Steve Parker, who's there third from the left is no longer in the lab, now an assistant professor at the University of Michigan. So this has been an empowering experience for lots of us who are trying to figure out how to connect things like the genome with gene expression, with genome-wide association studies showing risk factors for disease. How do we put this all together? Just today, Science Magazine publishing 3 papers on GTEx, this genotype tissue expression pilot analysis, looking at many different human tissues and understanding, therefore, how it is that genotype variations result in gene expression and EQTLs. And there are 3 other papers in other journals also out today as a simultaneous sort of first taste of what GTEx is going to produce, because this project will go on and build a greater power and greater numbers going forward. And I hope all of you are having a chance to begin to look at some of this data as well. It finds its way into projects that save graduate students and postdocs an enormous amount of time of having to do some of these things themselves, and that's always what we've been trying to do with genome resources of this sort. This, of course, has put us all in the enviable but sometimes vexing position of having a hell of a lot of data, and trying to figure out what to do with it has, in fact, turned out to be, at NIH, one of our major challenges, and one of the challenges that we want to be sure not to miss. So the big data problem has enabled us to bring together a lot of experts and make advice to us about what we should be doing, resulting in an entirely new project called Big Data to Knowledge, which is ramping up to about a $100 million a year effort. And this is something you can read about more if you haven't already read about it, because it's going to be, I think, something to watch if you're interested at all in big data sets and what to do with them. So it's a 6-year initiative at the moment, but there will be something, I'm sure, that will be needed after that. You can read the parts of it that are currently represented. Certainly includes training, analytical methods, sharing, but particularly centers of excellence to address biomedical analytics, computational biology, medical informatics. Those have already been funded, the first round of them, and we're counting on them to rise to this challenge. Of course, one place that we are all familiar with that's handling big data in a very big way is NCBI. The numbers here are pretty staggering when you look to see what they are dealing with. 10 terabytes a day coming in, 40 terabytes a day of data downloads going out. That is a phenomenal upgrade over what has been possible just a few years ago, and there's certainly no sign that this exponential curve is about to flatten off. Recognizing that, and that challenge is not going to get easier, we are actually taking a hard look at NIH in terms of the place where NCBI is located, the National Library of Medicine, and trying to imagine what should the future pathway be over the next 10 years for this part of NIH. And so a working group of my advisory committee has been established to try to envision what the future ought to hold for the National Library of Medicine over the next 10 years. And they're going to submit a report relatively soon, in about a month, which will guide us in terms of what we think ought to be done with this very important part of NIH. Well, now I want to turn to precision medicine, because a lot of people have been asking questions here at Cold Spring Harbor about this, and it's something that is very much consuming the time and interest of a lot of us at the moment who are trying to respond to an opportunity. And I want to tell you a bit about this, and also to encourage you to put your own ideas forward, because there are going to be multiple opportunities as I will go through to do so, including this meeting. It's not a new idea in the United States to try to form some sort of large-scale prospective cohort. Other countries, like the UK, have done this. The US has a number of cohorts that have been formed in various places, Kaiser Permanente, Intermountain, Mayo, Marshfield, Geisinger, some of whom are represented at this meeting, but we've not had a national effort. Some of us had hoped for that. Some time ago, I wrote this article. Actually, this is 2004, trying to make the case for such a prospective cohort. And, well, let's just say that's kind of what happened. It landed with a thud because people considered that to be utterly unrealistic and unaffordable, and that was probably true. I mean, after all, if you were contemplating a cohort where you were going to actually do significant amount of genotyping or even whole genome sequencing, as well as trying to capture the medical experiences of those same individuals, you would have wanted to have electronic records, which in 2004 were not present for most patients. And you would have had a number of other barriers, as you can see in this table. But look at what's happened here in the course of that 10-year period. So recognizing that, the momentum behind the idea of trying to do something of this sort has begun to build, and it reached the ears of none other than the President of the United States about a year ago. I will confess to have some role in how it reached his ears, but not the only role, because others have also been speaking to him about this. And he has become increasingly excited about the potential here and wanting to use his role as the scientist-in-chief to basically encourage some action here as opposed to waiting for this to take a longer time to develop. And so a number of meetings were called. Here's a photograph from one of them. This is in the Oval Office. You can tell we are all listening very intently. That is what you do when you're in the Oval Office and the President is speaking. And you may recognize at least one other person there, Eric Lander, who's the chair of the President's Council of Advisors on Science and Technology. But also in the room is the Commissioner of the FDA, the Secretary of Health and Human Services, the head of the Office of Management and Budget, and the head of the Office of Science and Technology Policy, the White House Science Advisor. And this was a meeting in October where the President basically had reached the point after much briefing from all of us that he felt it was time to do something. And of course, doing something means making an announcement, and timing is everything. And so the timing that was chosen was actually January 20th of this year.
So tonight I'm launching a new precision medicine initiative to bring us closer to curing diseases like cancer and diabetes, and to give all of us access to the personalized information we need to keep ourselves and our families healthier. We can do this.
Now, those of you who aren't from the United States are wondering what's going on here, but let me tell you, there was only twice in that whole address where everybody stood up, because usually it's one half or the other half, and you don't see them doing this in synchrony. So that was a good sign that maybe this was not just an idea, but a bipartisan idea that would have some support. But of course, nobody was quite sure what the idea was from that brief sentence or two. Harold and I wrote this piece that appeared 10 days later in the New England Journal. And if you want to follow from NIH's perspective where this is all going, we now have this website which is being regularly updated with activities, and there are a lot of them that are going on, because the President basically proposed that this would be something that would start next fall, fiscal year 16, which for the US starts in October. So what is it that we're talking about here? Well, let's be clear. Precision medicine is not a new concept. If you, after all, decided to get a new pair of glasses, you would be unhappy if that was just a one-size-fits-all. You want it right for you. If you need a blood transfusion, you probably want to be assured that some typing and crossing got done. But for most of the things that we have been doing in clinical medicine, we haven't had the information to be able to do that kind of precise approach. And so oftentimes physicians are stuck in the situation of having to give a recommendation that has been developed for the average patient and hoping that it will work. If you're lucky, oftentimes the recommendations are based on no evidence at all, but at least in some instances there is evidence, but it's generally not focused on the individual. But maybe the time is right to do something about that. In fact, I think one of the reasons to tackle this now is this coalescence of opportunities that come from technology. One is, of course, DNA sequencing, but that's not the only one. There's also the advent of electronic health records. There are the development of all kinds of wearable sensors that allow you to monitor various people's physiology as they go about their daily business. I'm wearing a Fitbit. I wonder how many of you are wearing a wearable sensor of some sort? Let me see some hands. Okay, it's coming. And there's a lot more coming, Apple Watches and so on, that are increasingly going to have sophisticated abilities to track how your body is performing, what your level of health is, maybe measure your blood pressure, assess the environmental exposures that you're encountering in real time. That is a great opportunity to begin to make sense out of gene-environment interactions. Furthermore, it's not just genomics that's coming along. You can now, if you want to look at metabolomics, sample hundreds of different molecules for a few $100s in a way that we have not previously had the opportunity to do. So these opportunities are coming along. This was, of course, produced in a very organized way in terms of an advocacy position by this IOM report, which is where the term precision medicine largely emerged. We'd been talking about personalized medicine before that. The IOM panel felt that personalized sounded a little bit too much like you were going to make a drug for each person. And that wasn't quite going to be practical, so it's more the idea that you're going to make a precise decision for each individual. I'm not sure I quite get it, the difference, and I think it will be fine if we use both terms and we'll all understand what we mean. So what's needed now to basically take a hypothetical and move it into reality? Well, we need a rigorous research program to provide rigorous scientific evidence, and that's not going to happen, just as it wouldn't have happened for the Genome Project, without talent. And so one of the goals here, one of the reasons I'm talking to all of you, is to try to recruit people to come and join from multiple different disciplines this gathering plan, which is still a nascent plan, as you will see, but it will get formed over the course of the next few months, and it should be a really interesting ride. So what's the vision? To do those things, we basically have two parts to this. There's a near-term part, which is basically taking advantage of the way in which precision medicine has advanced the furthest right now, which is in the area of cancer. We all can see the way in which these developments have been happening, much of it driven by genomics, but we could go faster if we made this into sort of the leading edge of this precision medicine effort. But in the longer term, we want to generate the knowledge base necessary to apply this to all areas of health and disease, not just disease, but also health, also prevention. So this is going to involve patient partnerships, because this kind of large-scale effort will fail unless the people who are involved donating their blood, their electronic health record access, are personally deeply engaged in the effort and sitting at the table. These are not going to be patients. These are going to be partners. It will have to involve electronic health records to be efficient. We'll need new technologies. Genomics will be a big part of what goes into this, and all of that will generate a lot of data science. So how do we put all that together? And by the way, the president proposes in FY16, starting in October, a budget increase for this on top of whatever else is available to NIH of $215 million. Most of that going to NIH. And you can see between cancer and the cohort, $70 and $130 million. So we have a lot of work to do to get ready about how we're going to plan to spend those dollars, assuming the Congress decides to go along. Remember, the president proposes, then the Congress disposes, and we can't be at all sure what ultimately will be provided. But we better darn well be prepared in case this is something the Congress embraces, which right now, I must tell you, they are pretty excited about. So let's talk about the near term. The cancer part of this. We already have, I think, a pretty good grounding to build this on between what the Cancer Institute has been planning, but we could expand those. The MATCH trial, which basically looks at solid tumors and lymphomas and basically picks for that particular tumor type the opportunity to do genomic analysis and then chooses drugs on the basis of what's found to be the driver mutations in those tumors. The Lung MAP program is looking at squamous cell lung cancer. There's an effort here to expand this to pediatric efforts, new cancer subtypes, a wide spectrum of cancers, early to late disease, and particularly to apply additional technologies that are nascent but perhaps can be scaled and tested in a larger way. One of those being the use of so-called liquid biopsies, not a term I'm particularly in love with, but the idea is to use circulating, oftentimes cell-free DNA as an indicator of what's happening in that cancer without having to stick a needle into it. There's clearly a need to understand when resistance occurs, why does it occur? We know that a little bit about that. We need to know a lot more. And maybe this is the one that, in my view, is most in need of studying if we're really serious about curing cancer, is how do you go from monotherapy to combination therapies, recognizing that monotherapy successes, dramatic as they can be, almost always result in a later relapse. And that is because there are so many tumor cells there that there's going to be some that are not going to ultimately be killed by your drug. All of those things are going to fold into the cancer part of this in ways that are not entirely yet clear, and with a timetable that's not entirely yet clear, and this is very much being led by the National Cancer Institute's leadership at this point. Just to sort of make this a little less hypothetical, I thought I'd try out a story on you. I tried this out on the senators last week, and they seemed to think, oh, that's what you're going to do. Okay, we get it. Well, it's probably a little simplistic for this audience, but just go with me for a minute here. If this was going to be successful, what would we be able to offer? So consider the story of Lily 3 years from now, who has a persistent cough. It turns out her exam's unremarkable, but her CT scan shows a small lesion there in the lung. We're not quite sure. She's had a bronchitis. Is this some residual inflammation? But a blood test, a liquid biopsy of various sorts now available or will be available in 2018, indicates that she's got an EGFR mutation, which sounds like she's probably got lung cancer. So the treatment in 2018 might involve surgical resection of the tumor, a molecular profile of the tumor. We can do that now. But then a combination of targeted drug therapies and immunotherapy, which is looking enormously exciting. But we still haven't really, in most instances, put together small molecule drug therapies, biological drug therapies with monoclonal antibodies, and immunotherapy. You usually get one of the above. And if that is then applied and gives a good response, she could have regular precision molecular profile of circulating cell-free DNA to find out whether it's working, and if relapse appears to be happening, to jump on that before things get into a more desperate shape. That's the kind of thing we'd like to encourage to become a reality for more and more people by the way in which this project would go forward. Okay, now let's talk about the longer term, which in many ways is tougher and more ambitious, because we want to basically design and implement a longitudinal cohort study that will go on for years and years and years and involve a whole lot of people. A whole lot, as in 1 million or more. So the idea here is we need to have abilities with such a longitudinal cohort to detect, measure, and analyze everything you can think of in an affordable way. Test out some of these in pilot studies first, and then use the promising approaches to extend them to the larger size of the protocol, and ultimately outside of this cohort to the practice of medicine. So this will comprise at least 1 million US volunteers. More would be better. And we don't have to start from scratch here, because there are, if you count them up, millions of people who are already in the United States enrolled in longitudinal cohorts that are being run by various organizations. But they haven't been sort of glued together in a way that would add to the power. Now that's not going to be trivial, because of course they have electronic health records, but those generally aren't very interoperable. They have different protocols, different consents, different agreements about the investigators, about how much data sharing they're willing to do. There's a lot of work here to figure out whether pasting these things together is going to be practical. We're in the middle of trying to figure that out right now. But undoubtedly, if you did that, you would not end up with a sample that was appropriately representative of the US population. So we will need to fill in those gaps. And we'll want to make this available to volunteers. I bet there are a lot of people in this room that would be interested in volunteering, and we hope to, by this fall, tell you how to do that so that people who are motivated and comfortable with the whole idea can be part of this national experiment. So participants are going to have to be centrally involved, as I said earlier. Otherwise, I don't think this will be acceptable. And they will need to get something for their participation. Information about themselves, information about the project, what's happening, what kinds of studies are being done, what's being learned. But I do think it's a new model for this kind of scientific research, and should be one that we can pursue and learn along the way about how to do it best. So another thing you might ask is, how is this going to give opportunities to learn things that matter about human health and disease that we can't easily do in other ways? And these are sort of the use cases that we ought to be thinking about before digging deeply into what is going to be a big investment over many years. So I want to try a few of these out on you. This is very much a non-exhaustive set of 5 examples, but I think maybe they'll give you a taste of some of the things that might be possible. I might say, when I was listening to the previous presentation, having 1 million people with genotypes and phenotypes available would be a pretty cool way to do Mendelian randomization of a lot of different things that you'd like to know more about. And that would certainly be on here, even though I have not labeled it. So first of all, simply developing new disease taxonomies ought to be possible when you have this kind of information about genes, environment, and phenotypes. You will undoubtedly learn things that tell you that the way we diagnose and label diseases is like way out of date for what it ought to be with the precision that we can now derive from this kind of information. I don't know if you saw this piece in Science from Tom Insel and Bruce Cuthbert, which is just a week ago, applying this question to mental illness. Right now, over there on the left, what we often do in terms of diagnosis is we use this DSM-5, and we basically categorize people by various symptoms. But I don't think any of us are very comfortable with the idea that that is a reflection of molecular understanding of the disease. If we put that together, and that's what people are trying to do with genetic risk score, brain activity, by various types of imaging, physiology, behavioral, life experience, we might then end up with a very different set of clusters, which would correlate more effectively with natural history and with response to therapy. So the idea of having this kind of million-strong cohort to be able to do that with any common disease, and certainly psychiatric diseases are plenty common, unfortunately, to make this possible, could give us that kind of taxonomic opportunity for change. Discovering therapeutic targets. Well, goodness, we've already been hearing at this very set pair of keynote addresses about PCSK9. There are other attractive targets that are emerging from the observation of individuals who have loss of function mutations and turn out to be actually protected against disease. That is your perfect moment to think about a new pharmacological approach, because it says nature has already figured out how to make an agonist, and it is both safe and actually beneficial. So SLC30A8 for type 2 diabetes is another example. NPC1L1, we saw that already on another slide. ApoC3, and of course PCSK9. And there got to be others like this, and a study of this sort would have the opportunity to uncover them. Identifying resilience is sort of related. I mean, there are people out there, probably, if you have 1 million genomes, and you have pretty good phenotypes on these people, who should be sick, because they've got some genotype that normally would be associated with a problem. They got a presenilin mutation, but the guy's 65 and he's got perfectly normal function and no amyloid in his brain. What's that about? CFTR mutations. You're going to find maybe people who are homozygous for CFTR mutations that ought to be causing lung disease, but they don't have it. An easy one, I'm sure, because most people with HFE mutations don't have hemochromatosis, but a few do. So why is it that the ones who don't? You can make your own list here. If you have this kind of very large database, you're going to uncover these protective factors. And again, those are going to give you ideas about therapeutics. And it's not all about genetics. You're going to find other examples of people who should have serious consequences of what they've been suffering, either as an illness like diabetes or as an environmental exposure like smoking, and maybe it'll give us a chance, because of very large numbers, to figure out how they're protected. Pharmacogenomics, in many ways, is sort of the first obvious way that one could use this million-strong cohort. If you already had all those genotypes developed and in the medical record, we could start to actually do a real-world test about whether pharmacogenomics helps or not. There are more than 100 drugs that on the label have something there about the physician might want to know the genotype before you write the prescription. But hardly anybody's using that information to try to optimize drug therapy because it's just too inconvenient. You want to write the prescription and you don't have time to send off a blood sample and wait 2 years or 2 weeks or 2 days for something to come back that you could then try to interpret. So mostly it's ignored. But if you already had that information, you could begin to actually find out for things like clopidogrel, for arterial clots, for warfarin, for antidepressants, for lipid-lowering agents. Make your own list here. All of those have drugs on them for which we know there are genetic variations that are associated with drug response, but they're not being used. We could find that out. And then exploring what works. And here I'm going to get more into the gadgets. The annual physical exam, if you look at the evidence that it actually does anything to improve outcomes, it's pretty flimsy. And partly it's because maybe we're just not collecting the right data. So you have that physical exam, you have some random blood tests. What's the evidence that those are actually good pointers to anything useful? I think, again, as we get into the space of being able to offer people more and more wearable sensors to keep track of their health, not just once a year, but in an ongoing way, we're going to have a better opportunity for health monitoring and health maintenance to actually have more effects of a beneficial sort. And that could apply not just to things like measuring your pulse or your blood pressure, but also medication adherence, tobacco cessation, prevention of diabetes by offering interventions to pre-diabetics, chronic disease management, and so on. We have not really tested out ways to do this that are now becoming possible, at least not at the scale this would do. So what are we doing? Right now, working intensively on trying to plan out the details of what this cohort will look like. I mentioned the cancer plans before, but the cohort is the intense focus now of quite a lot of people, with an advisory committee to me, basically a working group that has come together, co-chaired by Rick Lifton, and I'll show you the whole group in a minute. Broad expertise from lots of stakeholders, and they are charged with an interim report in September. We're working with the White House. The president wants regular briefings on how this is going. We'll do one next week. We're working with FDA and other agencies, and we need lots of input in order to be sure we're making the best choices about how to construct and design this long-term cohort effort. That's the list of the people who are leading this think tank that is intensively in the process of trying to give advice about this, and most of them, I'm afraid, are not going to have a whole lot of summer vacation. You will recognize some of the names there as people who are deeply engaged with this genome community. And you'll also notice that it's a mix of people between public and private sector. You may also notice the name Rory Collins there from Biomed in the UK, who obviously has some very useful experience to share with us about how that has gone across the pond. We're running public workshops. Ran one in February. We ran one just last week on how to basically put forward these use cases to be sure that we're thinking about all the things we'd want this cohort to do for us. That has been a very interesting experience. You'll see it was also tweeted rather heavily, just like this meeting is. And then over the course of the next 3 months, we will have 3 other workshops, topics on which you can see here, and not all of them at NIH, but some of them, trying to capture as much information as we can. These are public workshops. They're webcast. You can watch on the web or you can come to them. And they are basically an opportunity for this working group to seek input from the best and brightest about what we should be doing to design this properly. Well, maybe I'll just finish with another hypothetical about how you would take this cohort and somebody who was enrolled in it might benefit, and then how you would extrapolate from that to the nation as a whole. So here's Carla, who volunteers to take part in maybe in the fall, but by 2018, she's been receiving various amounts of information. She is otherwise healthy, but turns out she has an APOL1 variant that increases her risk for kidney disease, particular glomerulosclerosis, which is a common variant in African Americans, which turns out to be protective against sleeping sickness, but causes this risk in terms of one's medical health, especially if you're hypertensive. So she was offered the chance to try out one of these wearable sensors. At that point in 2018, there's a reliable way to do this for ambulatory blood pressure. Right now, that's not really quite the case, and does so, and unfortunately finds out she's not terribly happy with the result. She's walking around with blood pressures that are in fact high, and something ought to be done about that. So she gets on a diuretic drug, which, by the way, she checks on her own iPhone because she's also knowledgeable now about pharmacogenomics and makes us the conclusion that this is a drug that should work for her. And then she gets a smart bottle that basically notifies her smartphone if a dose is missed, because the big problem with hypertension is lack of compliance, and that's not just because people are being careless. It's just really hard to remember to take a drug every day, and so this is one more way to try to achieve that kind of compliance. She does so, blood pressure normalizes, Carla remains healthy. This is a good thing for that one person. But the real vision here, sort of stepping out from the research question, is could we, based on the experience of 1 million participants, establish in various ways with various kinds of technologies the value of precision medicine in a rigorous way, not just that we hope it's going to work, but that we have the data in prevention, in diagnosis, in treatment of disease, and then this becomes the standard of care, a standard of care which is accepted by the medical profession and by the agencies who decide reimbursement because it's actually shown to benefit outcomes. We need that data if we're ever going to go from where we are now, which is a very ineffective and wasteful healthcare system. So that's where we'd like to get to. And this is a big plan. I sort of come to this particular quote because it's one I used to regularly use when I was talking about the challenge of doing the Human Genome Project. And this is a quote from Daniel Burnham, who's an architect. But the Precision Medicine Initiative has that same flavor. It is not a little plan. It's a big plan. But little plans have no magic to stir men's blood and probably themselves will not be realized. So he says, make big plans, aim high in hope and work. Well, that's, I think, what once again we're trying to do with this effort. There are all kinds of ways that this could turn out to be really hard, and parts of it may turn out to be much more difficult than we anticipated. But it seems like the time is right. And if we have the opportunity to try to do so, in many ways, maybe at a somewhat late date in this country compared to other countries, then I think it's time to do so, and I'm happy to see that the President of the United States agrees, because that gives me a lot more of an opportunity to make it happen. So thank you for listening to all this. Again, if you want to read more about it, you can do so in that Precision Medicine website. You can read my blog every Tuesday and Thursday. I hope you are. Or you can sign up for my tweets, like many of you seem to be tweeting yourself. I want to be sure we have a chance to collect from you as much feedback as possible about this during this summer season while we're trying to design the project. So with that, I will stop and see if you have questions. Thank you very much.
Do we have a few questions for Dr. Collins? Yeah.
Thanks, Francis. Direct-to-consumer testing, how does that factor into the planning, and what are your comments, et cetera?
Well, actually, Ann Wojcicki, who's the head of 23andMe, was at our workshop last week, basically sharing what they have already done. They've enrolled now 950,000 people who have had genotype information obtained, and interestingly, their enrollments didn't even slow down after FDA told them they weren't allowed to give back any medical information. People still come forward, I guess largely because they're curious and interested in the ancestry data. She also says 85% of their participants say they are interested in taking part in research. If they're asked, they say yes. Now that's obviously a selected group. You don't sign up for 23andMe, I think, if you're not somewhat sort of motivated to pay attention to genomics. But it certainly says there's a lot of interest out there. Now, is that indicating that people want to have their own genomic information? I think it does. I think that's been well documented by many surveys. We're doing another survey right now through the foundation of NIH, asking people some of the same questions that were asked 5 years ago about their level of comfort about having other people see their genomic information in their medical records, but also what do they want to get back in terms of access to information. There is, of course, a very difficult conversation to have about what part of the information do you give back and what is going to be of more harm than good, because it's confusing and we don't know what it means either. And there will be no obvious answer to that other than every participant ought to have a chance to decide what they want. And if they've decided that, and it's clear that they've had the information to make that decision, we should honor that.
Just a question up here from Molly.
I was wondering what you saw the relative role of basic research versus more translational or applied research is in pursuing this.
Well, basic research is the foundation of everything we do, and it always will be. And certainly in the Precision Medicine Initiative, that's perhaps a bit more on the side of the applied part of NIH's portfolio. But again, even if this does get fully funded, this is 0.7% of the NIH budget. We spend about 53% of our budget on basic science. The basic science that goes in here, there will be plenty of that, particularly in the area of engineering, technology development. But the overall application here involving 1 million people, or the focus on cancer, will have more of a clinical bent than much of the rest of our portfolio.
Just up here, Francis, on the
So
top.
this is very exciting, but I didn't see any funding for training the doctors, because after all, they're going to be applying this information, and we're risking the situation where we have this underprivileged class of patients that don't have access to that, really.
It's going to be a challenge. It has been all along to try to come up with a system where physicians have the kind of information they need to be able to incorporate this kind of new digital data in a way that actually helps people and doesn't just confuse them. Again, I think one of the things that I'm hopeful about the model that we're following is that a fair number of the cohorts that we're trying to put together are actually healthcare delivery systems. Kaiser Permanente, take that as an example. So we ought to have a direct feedback loop between research results and healthcare delivery. And especially if it is a healthcare system that has a fair amount of diversity amongst the participants who are getting their healthcare, it provides a laboratory to see how we can shorten the time from knowing whether something is useful to actually getting it implemented, which has traditionally been a very hard problem. I'm not saying that's going to solve it, but at least it's a step in that direction.
Okay, we have one last question here.
Yeah, thank you, Francis. Just to sort of follow up on that, we live in complicated times and very political times, so I'm kind of wondering how you're approaching things like getting buy-in from healthcare delivery, health insurers, the pharmaceutical industry, et cetera, because a lot of those people have a lot of money to influence policy.
So that's one of the reasons we're holding these very public workshops and doing a lot of individual consultations with all of those stakeholders. I think so far this is a way of creating data sets that everybody's interested in seeing. Health insurers certainly are. Pharmaceutical companies are particularly intrigued about the way in which this might lead to new drug targets. They're also interested, if you have a cohort of this sort that's pre-existing with patients who have consented to be part of research, could they actually then approach those individuals saying, are you interested in a clinical trial because we see that you have this disease and there's no current effective treatment? And I think that might very well be something that could be done if the participants were willing to do so. So far, I'm not seeing a lot of objections. My main fear, of course, is that there will be no funds for this because, not to be a downer here, but the NIH has lost 23% of its purchasing power for medical research over the last 12 years. And to propose something of this sort in the face of that downward curve is perhaps a little bit optimistic. But on the other hand, if you ever want to turn that curve back upward, at least my experience in Capitol Hill is that you need to have something energizing, electrifying to propose. To simply say, we need more money isn't always going to be successful. Dr. Watson has a question or a comment
has
at
a
this
question.
point.
I need not deny him.
Francis, the last thing you saw, I just down the hill. NIH will get more money when we cure cancer. Until we do, there's a great deal of skepticism. When I read Newt Gingrich's give more money to NIH, I thought, oh, the study sections don't work anymore. There's complete, very hard to be innovative through the government system. It's almost enough to make you vote Republican.
Oh, not that.
No.
Well, on that note, I think before we all go off to a lobster, well, probably you want to, but you can't just yet because they locked the door. We're going to, I think we need a protest song, don't you? All right.
This is the genome guitar.
Yes, it is.
I think I know how to hand this to you.
Thank you. You did that very well. So yeah, and this is specifically constructed just for this occasion, and it is a protest song, and I'm ripping off a really wonderful song by Bob Dylan, which you will probably now wish I hadn't done because when you hear it in the future, you remember this instead. But it has a line at the end of each verse that needs group participation. Otherwise, I'm going to feel really frustrated, and I'll sing an extra 10 verses, and you don't want that. So I want you to sort of practice. This is The Times They Are A-Changin', okay? Some of you have heard that song. Some of you are as old as I am. I see you out there. Don't try to pretend you don't know this. So anyway, the last line goes, for the times they are a-changin'. You can do that, right? Come on. For the times that they are a-changin'. Okay, here we go. Come all you have gathered from far and from near. Let's all now agree that our goals are quite clear. Applying the genome's our greatest frontier. If it's the lives that we hope to be saving. So thanks to Jim Watson for bringing us here. For the times that they are a-changin'. I don't hear you singing, now come on. Come all you big thinkers, don't be surprised. As strategies newly are being devised, the pathways for progress must now be revised. The genome still needs functionating, and it's time to make medicine personalized. For the time they are a-changin'. Okay, it's going to be a political verse now. There's that thing called the sequester. Boy, do we need to get rid of that. Welcome, senators, congressmen, please heed the call. While payoff from research can be a long haul, with your help the science can run and not crawl, while America's the leadership saving, or China and India will just take it all, for the times that they are changing. Will it take a new Sputnik for insight to strike? Will we keep doing work like we did under Ike? Will science disruptions be met with dislike? And no end to damn sequestration. Hell no, we all shout, for the future is bright, for the times that they are changing. Well, the needs are compelling, the challenge is vast, and genome solutions are needed so fast. We're bold, we're audacious, not mired in the past. Our prospects are truly amazing. So come on, you people, let's go kick some ass, for the times that they are changing. Woo! Put up the slide. And come listen to the band at 9 o'clock out on the terrace. There'll be lots more rock and roll.